Welcome everyone. Today I'm going to present gene therapy. That's very interesting topic and a very new topic in biotechnology. There are some researches going on this topic, that is gene therapy. And now let's take start. In gene therapy, basically what we take gene from one organism or from one patient and replace it with another functional genes. It's basically replacement of defective genes in an animals in an living organisms. That is the basic idea of gene therapy. And if we talk about the mechanism, the processes which are involved in gene therapy, these are the vector mediated gene therapies, in which vectors are basically used to deliver functional gene and replace it with defective genes. These vectors have two types. One is viral vector and second one is non-viral vector. In viral vector, as name indicate, that it depend on viruses. We use the virus as a vector, and mostly adenovirus are used as a vector. And on non-viral basis, we use the liposomes. These are also used as a vector to deliver the genes. And if we talk about the types of gene therapy, there are two main types: in vivo and ex vivo. In vivo gene therapy, as name indicate, that in this we deliver gene inside the living body of an organism, while in an ex vivo we take out cell from the body and then we perform our experiment in labs. That is an ex vivo gene therapy. And now if we talk about the history of gene therapy, then in 1990, one of the patient named Ashanti De Silva had genetic disorder called severe combined immunodeficiency syndrome, SCID or SCID. In this syndrome, she basically has the deficient of ADA gene. This gene is involved in the accumulation of DATP, which is toxic to certain types of T cells. So it trigger and target the immune system of that patient. As a result, she became the immunodeficient for that disease. So in this gene therapy first performed on this 4-year old patient. In this we take out, as you can watch in this diagram, we take out this defective gene and we perform ex vivo gene therapy, and we use some virus vector clone with the ADA gene, and then it into a culture cells, and when they grow, then we take out these culture cells and put it in an human body. So that is the basic technique of gene therapy. Similarly, another example if we have, that is cystic fibrosis. That is the example of in vivo gene therapy. Cystic fibrosis is a lung disease. In this, the patient has deficiency of chloride ions in their tissue cells. So basically, there is a factor, regulatory factor, which is called cystic fibrosis transmembrane conductance factor, that is involved in the transfer of chloride ion. So in cystic fibrosis, due to abnormality in the functioning of this gene, this regulatory molecule cannot pump in these chloride ion. As a result, the patient became the deficient of that chloride ion, and which create a thick sticky mucus inside the lungs. As a result, it cause a lung disease. So gene therapy also performed for this cystic fibrosis, and it is in vivo gene therapy. That is the best example for in vivo. And here we have some data from one of the online website. I take this data. It shows the therapy clinical trials which are performed in different eras. These are in phase 1 60.3%, 18.9, something like that. And Uh, Another new technique which is evolving in biotechnology field is regenerative medicines. In regenerative medicines, they basically new cells and tissues are grown outside the body and then are replaced with defective tissues and organs. And this regenerative medicine, one of the main domain of this medicine is tissue engineering, in this replacement of tissues and organ by growing them in culture. It is basically cell based or scaffold guided technique. And here in this diagram I explained it in detail how this regenerative medicine technique is performed. Basically we take out these stem cells and grown outside the body, and then after this we uh, take out these cells and replace them with defective tissues and cells inside the body. And there is scaffold guided tissue regeneration. In this tissues we make a scaffold template. On this template grown cells and then replace them with the defective cells like this. In this example, this is basically a scar on a skin that is replaced with the new skin. And another application of regenerative medicine is bone regeneration, that is mostly performed and on a commercial level. The many experiments and many techniques are evolving for this bone regeneration. And another example is blood vessel regeneration, and last one is cell and tissue transplantation. In cell and tissue, we have many other types like autograft. In autograft transplantation, patients own tissue from one region of the body, like vein. If we take vein from leg and replaced with the coronary artery in a heart, that is performed in bypass surgery. And another example is organ transplantation, that is xenotransplantation, that is performed I think in 2021. American scientists performed this experiment. They take the tissues, the cells from pig, basically take the tissues of heart from the pig, and then they grow inside the body of a patient, and that patient survived for I think 2 months. After that, she died. But this experiment was successful, I think. That is xenotransplantation. In future, it has very big scope, and that's the two main basic types of tissue transplantation. One is autograft and second one is xenotransplantation. So that's it. Thank you very much.